Now that you know a bit about DNA, let's look at the key figures who truly made us know the facts behind DNA. December 15, 1916, Maurice Wilkins was born in New Zealand. He would grow up to be a kid who was engrossed in nature and super-duper adventuresome. He would later on credit some of his success from his early formative years in New Zealand and the influence of the exploratory early settlers of the time. In 1938, Wilkins graduates from St. John's College of Cambridge and earns a physics degree. At the time, England was at war, and therefore people like Wilkins were in demand. It is at this time that Wilkins works on improving the radar, and some of his work is actually still used today. In 1943, Wilkins's physics department was relocated to Berkeley, California, where he worked on the Manhattan Project. This was famously known during World War II as a group of scientists who produced one of the first nuclear weapons the world has ever seen. But because of the devastating effects of the bombs, especially in Hiroshima and Nagasaki, Wilkins became a strong opponent against nuclear weapons. After the war and in the early 1950s, Wilkins studied biological molecules like DNA and viruses using microscopes. He later on began to use X-rays to produce diffraction images of DNA molecules. These diffraction images produced by him, Rosalind Franklin, and Raymond Gosling did not succeed in mapping the structure of DNA, but the results were very, very important in Watson and Crick's eventual unlocking of the mystery, a long spiral with twin heads. He would later on marry to Patricia Ann Chidgey in 1959 and have two little darling kids, Sarah and George. In 1962, he received the Nobel Prize in Physiology or Medicine with Watson and Crick, and as an old man, take on the leisure activities of gardening and collecting sculptures. In 1920, Rosalind Franklin was born in London into an influential Jewish family, and at the small age of 15, made a bold decision to pursue science. As a kid, she would enjoy analyzing data and even did math problems for fun. In 1938, she was accepted into Cambridge University and there studied chemistry. After receiving her degree, she went on to study coal, which was very important at the time of war. Franklin later on moved to Paris in 1946 and studied how to produce X-ray diffraction images. So when 1951 came and she was studying with Maurice Wilkins in London, her X-ray diffraction images were much more mature than that of her colleagues because of her prior experience. What Wilkins and Franklin did was stretch a DNA strand across a paperclip that was mounted on cork, then send X-rays through this DNA strand, and their diffracted paths was then captured on sensitized paper. The X in the middle signified that DNA was in a helical shape. Franklin discovered two forms of images being produced of DNA. One of the pictures of Form B, known as Photograph 51, became famous as a critical evidence in identifying the structure of DNA. This image was produced through 100 hours of X-ray exposure from a machine that Franklin refined herself. Maurice Wilkins showed the image to Watson and Crick, who used this evidence heavily in their experiment to show the structure of DNA. When they finally published their revolutionary findings in 1953, there was no mention of Rosalind Franklin. She had no idea it was her research that led to this monumental discovery of DNA. In 1953, Franklin left King's College and was told not to study DNA. Therefore, she relocated to Birkbeck College, where she studied the structure of RNA, not DNA. In 5 years, Franklin published 17 papers on viruses, and her group laid the foundations for structural virology. She later died in April 16, 1958, from ovarian cancer at the age of 37. In 1962, Wilkins, Watson, and Crick all received the Nobel Prize for their findings without any mention of Rosalind Franklin. It is only until recently that Franklin's wide contribution is being credited. Therefore, she is known as the Dark Lady of DNA. On June 16, 1902, Eleanor McClintock was born. It was only until later on that her parents would call her Barbara because of her mature behavior as a kid. McClintock had a rough relationship with her mother, but on the contrast had a strong one with her father. So when it was the time, her father let McClintock go to college despite her mother's objections. In 1927, Barbara McClintock received a PhD in botany at Cornell. Afterwards, Cornell appointed her as the role of instructor in the botany department. It was at this time that McClintock involved herself in plant cytogenetics. In the early 1930s, both McClintock and her graduate student Harriet Cretan studied the behavior of chromosomes. After improving the staining techniques, McClintock and Cretan both proved the existence of chromosomal crossover. In 1936, Barbara McClintock became an assistant professor at the University of Missouri. It was here that she worked with X-rays and discovered the large-scale mutations and the chromosomal instability that can occur from the breaking, fusion, and bridging of chromosomes. Finally, in 1944, as visiting professor at Columbia University, McClintock studied the relationship between color patterns on corn plants and how their chromosomes looked. She really wanted to know why was purple spotted corn a thing. She discovered that parts of the offspring chromosomes seemed to be cut out and shifted to entirely new locations, hence the name jumping genes. At the time, genes were thought to be in fixed positions on the chromosome, but McClintock's research obviously negated this idea and brought up a new one of transposable elements.